Miss Danny, I can't hear you.
Hey, babe, I'm not talking yet. I'm just getting set up. You're good. Can you hear me now? Oh, now you're frozen. Ah, frozen. She's frozen. She'll be back. Hey, y'all. What's up? Good deal. You have a good fall break?
We didn't have fall break.
Didn't have any, huh?
No,
Had to work the whole time?
Yeah, our fall breaks from October 9th to October 16th. That's because that's when we're going on vacation.
Where are you going then?
South Florida.
Oh, nice. Okay, cool. Claire, can you hear now?
Yeah, I can.
Okay, cool. I got a couple others that are probably going to join in while we're doing that. I've got your Q2 packet. So if you have your, does everybody have their binder with them?
Yes.
Cool. If you have your binder with you, take it out. And I'm going to tell you exactly what to do. Okay, so what y'all need to do is I'm going to use as an example because Lily's not here today. So what you're going to want to do, pull this first. This is all the work I'm handing back to you until you get to the sticky tab. So if you think I'm handing back to you, put that wherever you normally put stuff. So what you want to do Great, I'll put those in the folder up there. Yeah, cool. All right, so wherever you normally put your work that's returned, which hopefully is probably a long lesson. Okay, and then after that, with those two? Sure thing. Sweet. All right, Sky, since you just joined us, we got the new Q2 packet, so I'm just showing them where to go. All right, when you go to your syllabus, Lily's in it. Your syllabus here, huh? Oh, I got Sky's, baby. I'm sorry. I grabbed yours. Sorry, Sky. I gave you such a drink. There you go. All right, so find your syllabus, and you're going to want to flip to where it says lesson 8, which is on page, a couple pages in, let's see, like 3 pages in maybe?
Don't you just put this in one of the syllabus?
This thing right here in the front that has all, hang on. So we're looking at this. Where's the camera? There. And you're gonna, when you get your new stuff, you're gonna flip to it, and where you have, that's okay. So page that has lesson 8 through lesson, this one that says lesson 17, you're gonna have to take that out and replace it with the new ones because they're updated. So all these ones that say lesson 8 through 17, so flip yours a little, flip, all right before Q2. So here's lesson 9, okay, 8. This is what we want.
Like this.
This right here.
Take all these extra
You
pages.
can take,
All these extra ones.
hang on, there, just hang on. Take those out, right there.
From here to there.
No, from 8 to 17. Yeah, it'll have this on the front, it'll say 8. On the back, it'll say Q3. That's where you want to end. And then you replace them with this new set. And then that goes in instead. So you take those out, perfect. So now you have all updated lessons.
I need to look for this.
Yep. You're
Do you
good.
take all of these out?
8 through 17, take out and replace. Because if you look, they all say like homework, TDA.
Why does this say Q3? No,
That's gonna be third quarter. That's okay, this is a new Q3. This is the new one. This is the new one that has, see, it'll say TDA. That means we have to be announced exactly what the homework is. So you want this new set. What?
Take these two out? I can't find the Q.
Oh, okay.
No, wait,
Everything from lesson 8 through the page that says lesson 17. All right, so this is the new stuff or the old stuff in here?
okay, so all of this.
Let's see. So where it says, like, see, it says homework TDA.
Yeah.
Now you're gonna know exactly what homework is. So we're gonna take this whole chunk out. And then where's your new set? Is it here?
No, that's it. Stuff that's in lesson 14.
That's fine. Through. There you go. That's whatever. Do whatever.
So you take these out.
Hold on, Drew, 1 second. These new ones are the old ones. Oh wait, this is, we want to keep Q1. Is this yours, Lewis? Hang on, Drew, I'll help you. Want to keep 1 through. What is this new one? Oh, what are we doing? Okay, hold on.
This is.
We want to keep the first ones. We just want to take out these. So these are accurate because that's Q1, right? That's our first quarter. And then this is where our second quarter starts. And now you've got these. This is the old one. This is the old one. This is the old one. So where are your new ones? Where'd your new ones go? Your new ones? Somewhere there's another one. No, we'll figure it out. Okay. Somewhere is your 8. I don't know where it went. Do you know where it went?
Plus an 8.
Yeah. This is the old one.
Lesson 8.
Yeah, that's the old one. That's okay. I don't know where your new one went, but it's online if you need it. All right, then everything else. Go just to the very back. And I'm coming. Go to the very back and put your rest of your lessons in. Put all the rest of your lessons in. This is all of second quarter. Take that orange one off the top because you have an orange one in the back, right?
Yeah.
So flip to here, open that up. Hang on to him for a minute. Put all these in.
How did you notice?
And then put that there, and that'll separate Q2 from Q3.
I noticed this morning there were. Okay, you take those 3 out.
What now?
Put those 3 out.
Yep. Next, Mr. Trash. Next, Mr. Put it back.
Tired this morning. Anyone else tired this morning? Yeah.
There you go. All right. We get it. What's up, gang?
I'm a bit lost.
We've seen a lot.
Okay, we're
Want
coming.
me to go to lesson 8 or the lesson 7?
You're breaking up, Claire. Say it again, honey. You're breaking up.
Want me to go to lesson 7 or the lesson 8?
We're doing lesson 7 today, but I was putting we're putting their new quarter in their binders, that's all. Okay. Well,
Oh, okay.
let's see if I have anybody in the waiting room. Nope, I'm good. Everybody's here. Billy may come later. All right, Drew. All righty, gang. So we're gonna go ahead and start our lesson. And I'm way over here, like 10 feet from y'all. So I'm gonna take this off so they can hear me clearly. Cool? Cool. All right. So we got, so if you're listening at home, you can mute your mic. Claire, because we get feedback otherwise. Thank you. All right.
Alrighty, let's see. All right, so we're going to start with our cell processes today. So we talked a little bit about some of the processes before, but we're going to talk a lot more about them today. So organization of matter. So everything in life is organized. And we talked before a little bit about how cells make up tissues and tissues make up what? Do you remember? Organs. Organs make up organ systems, and then systems make up, remember the last one? Organisms. Yeah, so yeah, it's been fall break. We've had a little break. We're all good. Okay, so matter itself is organized even more specifically than that. So we would start with this. We would say atoms, and that's the smallest unit of matter and has three main parts. Now, if we're going to be very technical, it's not the smallest unit of matter because it has three parts, and those parts are smaller than the atom. And there are even like what they would call, so your proton, neutron, and electrons are what we would call, you know, atomic particles, subatomic particles. And then you can get really tiny things that we can't even see, but physicists like to talk about things like quarks and yeah, quantum physics stuff. So keep in mind that this is pretty much all you need to know for biology, but it gets even smaller, right? So you've got atoms, and what are your three parts in an atom? Yep.
Oxygen, hydrogen, nitrogen.
Oh, those are the three. Yeah, those are the three represented there. What are the three parts in an atom, Drew?
Protons, neutrons.
Okay, what's a positive? Which one is positively charged? Wait, do you know?
Proton.
Yep. Which one's negatively charged? Electron. And which one has no charge? Neutrons. Good. You guys are so smart. All right. So keep that in mind because the way those are organized set up certain molecular structures to interact with one another. So after atoms, we have elements. So elements are pure substances consisting of only one type of atom. And it's they're identified by an atomic number. Y'all seen the periodic table before?
Yeah.
Like Drew was here last year for chemistry. It's really big and has all kinds of details on all these different elements that we know about. And then there's kind of even empty spaces for things that we know exist, but we haven't found yet, which is really pretty cool.
That doesn't make sense.
No, it does. And then there's also, we talked about particle accelerators where they put atoms in these giant, big circular underground tubes, and they race them around super fast till they smack into each other at such high force that they join and become a super heavy element, but they only exist for like a few seconds. So that's the fusion thing we're trying to do. It's pretty cool. So then you have molecules and compounds. Okay, here's the difference between molecules and compounds. Molecules are formed when any two atoms join together, right? But compounds, they have to be two different. So a great example is oxygen here. Okay, oxygen is never by himself. He doesn't like to be alone. So you'll never just find one oxygen atom floating around unless it's like quickly moving from one thing to another in a reaction. It's always with the friend. So if you have oxygen together, O2, which is what we see in the environment, O2, then that's going to be a molecule, not a compound, because it's just two of the same. But compounds over here is when you have more than one. And then there are lots of different types of compounds. And then there's also, not to be super confusing, but there are certain compounds that we consider molecular, and then certain we don't consider molecular, even though they're still compounds. So that's more for chemistry. But yeah, so atoms make up elements, elements make up molecules and or compounds. So when we talk about biology, we talk largely about carbohydrates. So carbohydrates are the most common, or the most common molecules, compounds on the planet. They are super, everything that has to do with biology is about carbohydrates.
My mom said I had carbohydrates in my breakfast, and I'm like, I know I did, but that's like not the type of carbohydrates I'm talking about.
Yes. So you'll hear people talk about them when they talk about diet nutrition. They'll talk about carbs, right? And eating too many carbohydrates. But there are different types of carbohydrates. But whenever you hear anything about carbon-based life forms, we're talking about carbohydrates. Or when we talk about organic chemistry, you're talking about carbohydrates. Anything organic contains Carbohydrates.
Starch is
Yes, starch is a carbohydrate. So here are some types.
you cannot live without carbs.
Yep. So here are some different types of compounds. So here are some examples of carbohydrates. So you've got monomers, and sometimes they have subunits. So these are things like sugars, okay? This is a monosaccharide. So this is a simple sugar. Then you have things we call polymers. See, monomer, what does mono mean?
Singular.
Yeah, and what does poly mean? Yeah, a lot. So can you see how this has one little ring and this has tons of those little rings stuck together like a chain? So this one's a monomer and this one's a polymer. So polymers are things like starches. Starches and polymers have very long, their compounds are very long chains. And then here's another example. The granules that are in chloroplasts inside chlorophyll, inside your plant leaves and plant cells, contain carbohydrates. Then we have nucleic acids. Okay, this is another type of compound. So this is a nucleotide, and this is what DNA looks like, right? And it's these nucleotide bases. So they would be the purple, pink, green, and yellow bars located here. That's what they're representing. Those are nucleotides. And then you would find all that inside a chromosome. So basically, it's like one of these put together makes one of these, and you'll find them in here. And one of these put together with others makes this, and you'll find that in here, right? Then we have amino acids. Okay, so amino acids actually come, as we're going to learn, from the combinations that are coded for in DNA. Okay, and there's like 23 of them. And you can, it's like certain Legos. It's like if you had these particular Legos, you could make nearly an infinite amount of structures with them. And that's how it works. So you get these amino acids and they go together in a chain. This is called a polypeptide. And the reason it's called a polypeptide is because amino acid, the bonds in here where these little bars are, those are called peptide bonds. So they call it a polypeptide. And here's an example of one. So this just says it's an intermediate filament. Okay, so probably something like maybe like a collagen or something that's in your hair, something like that. Now, fatty acids. Okay, fatty acids are the lipids. So lipids are the other type of compound. These are the things that are really important in living things. And a triglyceride would be an example. And you can see why would it be a triglyceride, do you think? What do you notice about it? What, Drew? Three chains. 1, 2, 3. That's exactly right. And this says adipose cells. Do you know what adipose cells are? Anybody know what adipose cells are? You want to guess?
I'm going to go with cells close together.
Kind of, yeah. So pinch right here, anywhere on your belly. You feel the little fatty cells that are right there? They're there to insulate your insides. Those are adipose tissue. It's your fat tissue that you're supposed to have. Some people have way too much. It's actually yellowish in color. If you've ever seen like pictures of a surgery or whatever.
I once picked the scab so much it actually got down to the tissue.
So above so you've got, skin, your epidermis, and you get down into your skin. And then you have this layer of stuff called fascia. And it's more like these filaments. It's like packing tape. And it keeps everything together. And then underneath of it, you'll find stuff like fat, or fat can be on top of it too, depends on where you are. So we've got carbohydrates, and they break down into two main groups. And those are going to be simple sugars, or what we call saccharides, or complex sugars, or polysaccharides, right? Complex would make sense, more than one. Simple would make sense, one, right? There's that poly word again. And then the simple sugars break down into the monosaccharides, which we talked about before. And then this one is fun, oligosaccharides. Now these are just like super big ones. That's basically all you need to know about that. And then we've got homopolysaccharides and heteropolysaccharides. Don't need to worry about that. Just know they exist, but know that right here, do you see this where it says starch and cellulose? Okay, so cellulose, if you remember, that is what makes up cell walls in plants. And it's what we call what? What do we call it commonly? Do you remember? It's outside the membrane, but we call it something like in diet, nutrition, and cooking. We call it fiber. So when you talk about eating something that's high in fiber, you need more fiber in your diet, it's just plant cell walls. That's all it is. It's cellulose. And we cannot digest it. Now, can you think of an organism that actually eats wood? Yes, Darcy. Termites. Okay, so here's a question. Do you think termites can digest cellulose? No. Okay. What do you think? What do you think, Lewis?
Beavers.
The beavers. Yeah, they chew on it. They don't swallow it though. What do you think, Drew? They can't. It's right. Both of you all are right. So they eat wood, but they can't digest it either. They actually have microorganisms in their gut in a tiny little termite digestive tract that actually break down the cellulose because they can't break it down either. Interesting. So then we have proteins. All right, now here's what I want you to notice about the proteins. First off, they're long and they go funky angles. These are just particular types of models of molecules and compounds. There's many ways to model molecules and compounds. But what I want you to notice is that some of these have what it says here are electrically charged side chains. Okay, here's why that matters. This one right here, arginine, see that it has a little positive down here? Okay, that means that right here at this end of the molecule, this model of the molecule, there's a slight positive charge. It's usually like a +1 if we were trying to like balance them, okay? And this one too. And then these right here, like aspartic acid or glutamic acid, they have a negative. All right, now the reason that's important is because these guys and these guys, what do you know about magnets and positive and negative? Like, what do you know, Darcy?
If you put the two same sides together, they'll like try to push away.
Mhm.
Like, if two different sides together, they'll like attract to each other.
And the same thing happens with molecules and compounds. Exactly the same thing. So it will allow for bonding, it will allow for interaction, and all that is super important. If our bodies didn't have this type of tiny little one extra electron, basically, a lot of the time it's 1 or 2 extra electrons. That's all you need so that these particular molecules can interact with one another. That's a big deal. That's a really big deal. So here's some examples of types of proteins. We have tons of proteins in our body and they're made up of amino acids. And next couple of weeks, we start talking about DNA and inheritance and how the information in DNA codes to make an entire organism. We're going to talk about how proteins are built. And it's really crazy because it comes down to those little tiny amino acids and that DNA code telling them exactly what to build. So different types of proteins, you have some that do structure. Okay, so a good example here is it talks about spider webs. Everybody has seen the JrLRCL webs now because we saw them when we were out there. And have you seen the power lines in this county lately?
Apparently they're invasive.
Oh yeah, they are.
Yeah.
Very. Yeah.
That's why I'm gonna bring
Wait, Drew, just-
water guns on my hike.
Yeah.
They say fill them with salt, squirt them.
Yeah, their exoskeletons are pretty hard. So I've heard that they don't absorb the salt as well as some other insects may. But I have heard that spraying their webs can actually have an effect because very often they eat their own webs, like they're cleaning up. A lot of spiders do. I think JrLRCLs might, but they're going to have to come up with a solution because they're dangerous to our invasive species. And you can't drive down any road in this county without look at the power lines on your way out of here. I'm not kidding. There are strips of power lines where it's just one orb after another. It's crazy. So a good example would be spider webs. Another one's collagen. A good example would be spider webs. Whoop. Somebody's not
on our tree in the front yard. We have one and then one in the attic. So we named the one in the attic Roro and the one on the tree Jojo.
Jojo and Roro. That's great. So another great example is collagen. Okay, collagen. Where do you find collagen in your body? Do you know? You know?
In your face.
In your face, in your hair, your nails, and your skin. And in your bones too. Yep. Well, that's cartilage. Yep, cartilage. Yep, there's collagen in there. So he's been out partying again and doesn't know what time it is. That clock, I'm telling you. So that cuckoo has issues. I think he goes out and parties and then comes home and forgets to do his job properly. That's what I say. So anyway, yeah, your hair, your nails, your skin. All right, and that's what feathers, things like that on birds. So then we have storage proteins. Okay, so ovalbumin is the one that's inside of egg white. And it helps for the developing embryo. That's where the little developing chick or bird or whatever gets what it needs in order to grow. And then there's casein in milk, and that's important for baby mammals. Now it says plants have storage proteins in their seeds. Why would that be important, do you think? Why, Drew?
Storage in their seeds.
They have storage proteins in their seeds.
Because you
need energy, you need protein in order to grow it.
Yeah, exactly. It's gonna hang on to what's necessary until that little baby plant is ready to grow. So then you have transport proteins. So these are the ones that you're gonna find. Some of them are going to be in cell walls, and they're going to help transport things in and out and across a cell wall. And some of them are things like hemoglobin. When you go to the doctor, you ever gone and they do the little finger prick? Okay, so what they're checking for is they're checking hemoglobin, but they're doing it by checking red blood cells. And the reason that's important is hemoglobin is almost like a basket, that the shape of it, and it actually holds oxygen molecules. So if you've ever looked at a red blood cell, it's almost like a donut where the hole didn't go all the way through. It's like somebody took a ball and squished it on both sides like this. So it's round like a disc, but it has a big depression in the middle. And it actually holds all the oxygen molecules there. And as those red blood cells move through your body, they oxygenate all the rest of your tissues. And that's what hemoglobin does, is it holds the oxygen. Yeah, Louis?
It must be basically like a donut ball.
Kind of, yeah. Yeah, kind of like a donut hole, but if you'd squashed it even in the middle. So then you got hormonal proteins. So your hormones are actually proteins, and hormones aren't just like, oh, teenage hormones. Hormones are things like insulin, which you need to keep your blood sugar levels regulated. Diabetics have issues with insulin very often. And also, you know, everything that helps you grow male or female, those are hormones. And then there's other hormones that, you know, help you react in certain situations. There's all kinds of hormones. Hormones are basically just communication molecules in your body. Receptors. So these are things that respond to chemical stimuli. So built into membranes of nerve cells to detect chemical signals released by other cells. So that's important, obviously, in just sort of your general communication network in your body. Now, these are some of my favorites. Contractile proteins, actin and myosin. All right, actin and myosin are freaking cool. And when we get talking later in second semester, we get into, we're going to get more deeply into anatomy, and we're going to talk about muscles and things. And actin and myosin are two particular proteins that function inside muscles. And the way they're shaped, it's like one of them is kind of shaped like a cup and the other is like a little hammer. And it, the way your muscles contract is there is a reaction that pulls the hammer down, and it kind of moves to the next one, and then the next one, and then the next one, like a ratchet. If you've ever seen how a ratchet works, it's exactly the same way. And that comes down to little tiny proteins and the way they're shaped, that they ratchet inside one another and they lock and then they release. So that's how you can hold your muscle here and then release it. It's because they've locked. So what's going on is itty bitty, tiny ratchets in all your different muscle fibers. It's pretty neat. Defensive proteins. So these are your antibodies. So this is what you're hoping to build if you catch, if you catch COVID, you've hopefully produced antibodies to it, or if you've gotten a vaccine, you've been given antibodies to it. That's what those are. And lastly, enzymes. And do you remember what enzymes are? Anybody remember? In particular? Enzymes are catalysts found in nature. So what's a catalyst? Do we remember what a catalyst is? You know?
Something that nature produces.
Kind of. So enzymes are the natural version of catalysts. Do you remember, Drew, what a catalyst is? So a catalyst is a chemical that speeds up a reaction by lowering the activation energy of a chemical reaction. Enzyme is a catalyst. Yeah, an enzyme is a natural catalyst. So here's our lipids. All right, so with our lipids, we have these four main types. So you've got triglycerides and phospholipids. Didn't mean to do that. Steroids, and then waxes. Okay, so I want you to look at this guy, phospholipids. These are super important because this is what make up a cell wall. Do you remember when we looked at what that cell wall looked like week before last? And it had the little heads and tails? Well, there they are, right? And then also you've got triglycerides. You can find them in fats and oils. And then, and they're in your body too, obviously, because you have fat in your body. Yes.
The last slide, I thought it said liquids.
Oh, liquids. Lipids. Yeah. And then you've got steroids. So steroids are things like cholesterol, and cortisone is a great example. So cortisone is something that we often use in medicine. If you've ever been like really sick and you're just not feeling well, or you've got some inflammation that you can't kick, your doctor might give you prednisone. You ever had prednisone? That's cortisone or very similar. Or you might get like a cortisone shot. Some people will get cortisone shots like in their joints if their joints are really sore. There's all kinds of uses for it, but they will reduce inflammation very, very quickly. And then waxes. So waxes found in nature or created by nature, things like plants. Some plants have waxy leaves. You ever seen the plants with the waxy leaves?
Yeah.
And then what's in your ears?
Yeah.
Wax. Yep. And then you've got, like, think about honeycombs, beehives. That's wax too, right? So here is a video for you, which shows you how cholesterol and cell membranes all work together.
Welcome back to my channel. In today's video, we're going to look at the structure of cholesterol and the function of cholesterol in the cell membrane. Cholesterol is found throughout animal cell membranes. It is not present in the cell membranes of plant cells, as they have a cellulose cell wall that helps to support the cell membrane. Cholesterol is a lipid that is identifiable by its four-wing structure. It consists of four linked hydrocarbon rings forming the steroid portion of the molecule. Extending from one end of the steroid is a hydrocarbon chain. Extending from the other end is a hydroxyl group. Cholesterol is amphipathic, which means part of it is hydrophilic and other parts are hydrophobic. The steroid portion and hydrocarbon tail are hydrophobic, whilst the hydroxyl group is hydrophilic.
Do you remember what that means? Hydrophilic
Hydrophilic.
and hydrophobic? Hydrophilic means it is attracted to water, and hydrophobic means it repels water.
The cholesterol sits in this membrane with the hydroxyl group, which is polar and hydrophilic, in with the polar and hydrophilic phospholipid heads. The hydrophobic steroid rings and hydrocarbon chain portions of the cholesterol molecule are found embedded in the hydrophobic portion of the membrane with the phospholipid fatty acid tails. These cholesterol molecules carry out an important function in the cell membrane. They function in both regulating the fluidity of the cell membrane as well as regulating the permeability of the cell membrane. The location of cholesterol between phospholipids helps stabilize the membrane, giving it additional support in the absence of a cell wall. This stabilization allows the cell membrane to remain fluid but reduces the fluidity overall of the cell membrane. In addition, the presence of cholesterol between the phospholipids prevents the phospholipids from coming into contact, which prevents them from stiffening or crystallizing. Cholesterol's presence in the cell membrane is also in regulating the permeability of the cell. If the cell membrane is too fluid, the permeability of it would be too much, which would allow too many molecules to pass into the cell. If it was too stiff and had too little fluidity, not enough molecules would be able to pass through into the cell. Thank you for watching. If you want to support this channel, please subscribe and check out more of my videos.
She feels super tired. Like you walked out.
Yeah, she does. She talks really fast. I think she's
She just
I think she's from New Zealand. That's my guess. My guess is New Zealand. It doesn't quite sound Australian to me. I could be wrong. It's one or the other though. They're pretty similar.
Welcome back.
Nope, we heard you. So if you've been through these, let's look at them. So what is the most compounds that contain carbon? Which of these would be our answer for A? What do we call most compounds that contain carbon are what? They're going to be organic. Oh, sorry, Drew. Didn't see your hand. All right, how about this one? Type of nucleic acid that plays an important role in the production of proteins. You got a guess, Drew? Let's see. RNA.
Oh, yeah.
Yep. Now we're going to be getting to that really involved in that in the next couple of weeks. All right. Compounds that do not contain the element carbon. Yeah, Drew.
Inorganic.
Yeah, that's right. So if it's got carbon in it, it's organic. If it doesn't, it's inorganic. So when people say they're eating organic, they don't really mean they're only eating things with carbon, because that's really all you eat anyway. They're just trying to say that they're not eating things that have been fertilized with
No, they're just
inorganic
trying to say that
compounds.
they're better than you.
No, that's Drew. Stop. Don't do
They
that.
said they're better than
Hey,
you.
Drew, stop. Okay. All right. Thank you. So let's see what's next. Where are we? Okay. type of nucleic acid that passes from parent to offspring and directs the cell's functions. Ooh, Darcy, do you have a guess?
It's either molecule or nuclear acid.
Nucleic acid. Let's see. It's going to be DNA. Ah, there we go. How about very large organic molecules made of carbon, oxygen, hydrogen, nitrogen, and phosphorus? Nucleic acids. How about large inorganic molecule? I'm sorry, large organic molecule, duh, made of carbon, hydrogen, oxygen, nitrogen, and in some cases, sulfur. Yeah.
Amino acid.
There we go. You got, what would I just click? Nope, nucleic acids. I clicked it, but it didn't go. All right, and then how about, did I click that too accidentally? My finger gets excited. Smallest unit of most compounds. Lewis.
Enzyme.
Enzyme, what do we think? Smallest unit. Think back to that first image with the, this is made of this, which is made of this.
Atom.
Yeah, Lewis.
Atom.
Atom, yeah, Claire got it too. Atom's good. Oh, wait a minute, that's in the wrong. Did I just hit I? Where's G? G is down there. Oh, wait. So wait, yep, sorry, I read them backwards. The smallest unit of most compounds is a molecule because it's usually two stuck together. But the smallest unit of most molecules, when we get to that, wherever that is, or smallest unit of an element, we're going to get to atom down here. Sorry, I'm confusing y'all. Where are we? HI, the chemical combination of two or more elements. Two or more. Compound. There we go. And then how about type of protein that speeds up a chemical reaction? We just talked about this. It's a natural catalyst. So that's going to be an enzyme. There's your J. All right. And then how about smallest unit of an element? That, the smallest unit of an element. That's going to be your atom. There you go. And then lastly, any substance that cannot be broken down into simpler substances is an element. All right. So carbohydrates. What's an example of carbohydrates? Did anybody put anything? What do you think? Yep, go ahead.
Wait, no, sorry.
Nope.
I thought I knew that. I didn't know.
So sugars, starches, that's what we're going for. What do we call fats? Do you remember what the word was that would go here? Yeah, do you remember?
Fatty?
Those, well, lipids, right? Lipids are fats and fatty acids. So yep, okay. And how about, what are we gonna call, what's gonna go here? Does anybody know? Oh, sorry, wait, here, we gotta go here first. Help form cell membranes, and what else do lipids do? Cholesterol is what's going there. All right, so enzymes, what are they? They are gonna be proteins. All right, and then lastly, this one should be easy. We did carbohydrates, lipids, proteins. What's the last thing we talked about? Some kind of acid, what was it? Nucleic acid. It's going to be your DNA.
I keep wanting to say nuclear.
I know, and they direct all the cell's functions. So that would be DNA and all your kind of genetic materials. That's what would go in those. All right, so the cell and its environment. I'm going to try and speed up a little so we don't run out of time. So we talked about this phospholipid bilayer, I think a little bit week before last, maybe. So you have your cell membrane. It's selectively permeable, which we saw with our experiment, right, with the iodine. All right, and that means that it allows certain things to come through and certain things not. It allows certain things out, but not in but not out, all that kind of stuff. You guys know that. Now we got two different ways of things getting in and out. We have active transport and passive transport. So passive transport is what we would call diffusion. And this was, did you guys see, I posted a little video or actually some slides, I think. So with Lab 5, one of the things that people had the most trouble with was this understanding of diffusion. So basically, when you have a membrane, it's trying to get homeostasis on both sides. It's trying to get a balance on both sides. So if you have more particles of something on one side than on the other side, they're going to want to go through. So if they can go through, they will go through. And that's simple diffusion. And then that's passive transport. So that's basically simple diffusion permits passage of only small non-polar molecules across the plasma membrane. Drew, do you have a question?
Passive means it requires no energy to pass through.
Correct. Active means it does. Yes. So we also have what we call facilitated diffusion. Okay. So facilitated diffusion is where you've got passage of larger molecules through the membrane. and they can pass in both directions and opposite of a concentration gradient. So your concentration gradient is how many particles you have here versus here, okay? And you want, there's a membrane separating them, it's gonna want a balance always. So passive transport, the best example is what we call osmosis, and this deals with water specifically, okay? So water passing through one membrane or passing from one thing to another thing to keep a balance of water, that is osmosis. So it's very important, for example, with blood cells. So there's something we call tonicity, and it's how much tone, or basically like how, like if you push on a balloon, how firm is it versus how soft is it? So hyper, what does hyper mean? Do you know?
Hyper means very energized. Also,
Mhm.
that one that's Kaito.
Cytolysis, yeah.
It looks like a whoopee cushion.
It does, yes. So hyper would mean too much, right? And hypo means too little. All right, and iso means equal. So this is what your red blood cells are supposed to look like. Remember when I told you they look like a little donut where the hole wasn't completely punched all the way through? Or like a little ball that's been squashed on both sides. Okay, so if this is in an environment where there's too much salt on the outside, for example, let's use salt as an example because sodium is a great example. Too much sodium on the outside of the cell membrane. It's going to pull too much water out of the cell and the cell's going to shrivel up. If you have too little sodium, it's going to, if you have too little sodium, if you have too little sodium outside, okay, let me clarify. If you have too little sodium outside the cell, it's going to put more sodium inside and more water out. And in this case, you have the opposite thing going on. This is when you have too much sodium outside the cell, which, inside the cell, pulling water into the cell to balance it. So then it swells up. And if it swells up too much, we have cytolysis, which literally means bursting open. Dead. So you've got osmotic pressure, that's to deal with tonicity. That's applied to a solution side, keeps movement flowing. And then hypertonic means it has a higher solute concentration outside the cell. And hypotonic means it's got a higher solute concentration inside the cell. And isotonic means it's the same. Remember, a solute is when you just anything dissolved in anything else. So in our example, it's like sodium in water. So aquaporins are integral membrane proteins that are help water move in and out of cells. So active transport proteins. We have some proteins whose entire job it is to move things in and out of cell membranes. And they form little channels like this, and they actually make little gates that use chemical reactions to open and close like a real gate would open and close to a tunnel maybe, okay? I gotta watch our time real quick, sweetie. So they act like a pore in a membrane and they let certain things through. And then there's some that are gated, right? The gated ones literally act like gates. They open and close. It's pretty cool. So we're gonna whip through this next things because I'm watching our time. So you got transport pathways through a cell membrane, basic mechanisms. These are just some examples. It's a diagram of what happens. All right, so for these real quick, water moves out of cells of a saltwater fish and into the ocean. That's osmosis. Oxygen moves from the lungs and into the bloodstream. That's diffusion. Sodium is pumped out of a nerve cell. That's going to be active transport. So how is osmosis different from diffusion? Osmosis always involves water. Compare and contrast active and passive. As Drew said, energy versus no energy. Active requires energy, passive does not. Two methods of active transport would be protein transport and engulfing, which is when a cell like literally kind of bites another cell. It's really cool. And state one reason that cells are small, well, because things have to move through the membranes. They have to be very, very small. So true, selectively permeable means letting some, but not all substances pass through. Osmosis, no. Diffusion is the process by which molecules tend to move from one area of concentration to an area higher, from higher to lower. The process by which water moves across a selectively permeable membrane is called osmosis, if it's water. The diffusion and osmosis are types of passive transport, right? And then active transport requires energy. So really fast, we're going to whip through this next little bit because we do have our lab to do. So heterotrophs and autotrophs. So you'll remember that autotrophs use inorganic energy sources like sunlight, right? So what are some autotrophs? What's an example of an autotroph? Darcy?
An autotroph would be
What uses sunlight to make
Oh,
energy?
plant.
There you go. Perfect. Plants are great examples. And heterotrophs use sources such as other living things as a source of energy. So an example of a heterotroph was
Lion.
a lion. Great example. Okay. And photosynthesis is the process that uses light to produce food molecules. All right. Chemical energy and ATP.
Why did that child have a sword back there?
I don't know. I don't know. Looks like a sword. I didn't even notice, but you're right, it does. Huh. Oh, okay. Well, then there's
Got
that.
a sword and a shield.
So ATP is the molecule we use to make energy. Okay, that's that's basically how we have, that is our energy molecule. And there's a whole process involved in this. And we're going to go over it briefly, but I don't expect you guys to memorize the cycle. Okay, there's a whole cycle involved in this where you have your ATP and it's the end product of adding a phosphate to a molecule called ADP. So it's adenosine diphosphate. You add a phosphate to it and it becomes adenosine triphosphate. And cells release energy by removing that phosphate group to go from ATP to ADP. And all living things use ATP. Cells regenerate ATP and ADP and only contain tiny amount of ATP at any given time because it goes so fast. So chlorophyll and chloroplasts. So what's chloroplast, guys? It's a pigment. Okay, so what color does it absorb and what color does it reflect? Think about it. What colors is chlorophyll? Yeah, what color is it? Green. So what does it reflect? What color light? What wavelength? Green light, and it absorbs all the other colors. Very good. So it absorbs all wavelengths of light except green, which it reflects. And it's found in thylakoid. And that's these little stacks here. The stacks are called granum. And then there's a thylakoid and it's this little disc. And stroma is the fluid matrix surrounding. So like in here, there's liquid. It's called stroma. All right. So I'm going to real quick, hold on. I'm going to come back to this one. I want to make sure that we don't run out of it. Nope, because it's a good video, but I'll have you watch it last. I'll do this first. All right, so CO2 plus H2O plus sunlight, what's that going to give us? Do you remember? This is photosynthesis. It gives us oxygen and sugar, right? That's sugar. So what raw materials are needed for photosynthesis? These two right here, right? What are the products of photosynthesis? That would be what does it make? Oxygen and sugar. Very good. So why is sunlight written above? It shows the energy input, right? And where does it occur? It occurs inside chloroplasts. So the process by which a cell captures the energy in the sunlight and uses it to make food is photosynthesis, right? And what are colored chemical compounds that absorb light? Pigments. In this case, we're talking specifically about chlorophyll. The main pigment is chloroplasts or chlorophyll. And that should say chlorophyll because it says found in chloroplasts. What are small openings on the undersides of leaves? Do you know what those are? The little tiny holes on the underside of leaves? Do you know what they're called?
I think they're cells.
They're called stoma. They're itty bitty little holes. That's what lets the CO2 in so they can have this photosynthesis reaction. An organism that makes its own food, autotroph, and cannot heterotroph. So real quick respiration. Ooh, we're running late today. So respiration is another very involved cellular process. It's okay though, because our lab is not going to be very long. So if we run a little late, it's not the end of the world. And there aren't very many people coming today. So yeah, well, we're going to actually probably change our groups a bit and it's fine. So respiration, we're going to come back and watch this one too in a minute. We're going to watch that as a group actually. So photosynthesis versus cellular respiration. All right, so when you're dealing with photosynthesis versus cellular respiration, they are opposite chemical reactions, exactly opposite. That's photosynthesis, right? Carbon dioxide plus water yields sugar and oxygen, correct? The exact opposite is respiration. So it's sugar and oxygen yield water and carbon dioxide. So they're exactly opposite reactions. And then, but they also are super intermingled with all this different energy production. There's something called Krebs cycle, Calvin cycle, gets very, very involved. You guys don't need to know all of that necessarily, but it gets very, very involved. So with this one, what we have is fermentation. Now, the reason I wanted to definitely get to this, because this is today's lab. So fermentation is another way of producing energy that isn't respiration and it isn't photosynthesis. So it produces, there's different types of fermentation. The one we're dealing with today is alcoholic fermentation. And that's because an ethyl alcohol molecule is produced. And this is what happens with yeast. In bread, beer, wine, it's alcoholic fermentation. And then there's also lactic acid fermentation, and that's because a lactic acid molecule is produced. And this happens in your molecules, in your muscles. If you overwork your muscles, they can carry out lactic acid fermentation. And sometimes, if you've been working really hard, like your muscles feel almost crunchy. That's what that is.
They make like that cracking.
Yeah, those are your joints usually, but your muscles can get lactic acid buildup in them, and it's not fun. So there's similar processes where you put in organic compounds, you have this same reaction with like glycolysis, you produce this ATP, but then this says aerobic pathway, which means it involves oxygen because it's respiration. And this one says anaerobic, it doesn't involve oxygen. So photosynthesis is process by which plants capture light, produce energy. Cellular respiration is process of producing ATP using oxygen. Fermentation, you make ATP, but you don't use oxygen at all. Here are your three formulas. And this one just lists that there are two types because then we'd have to squish a whole bunch of formulas in. So where does photosynthesis occur? In what? Yeah, plants inside of what in the plant? Inside of the what? Inside of the chloroplast in the cell, okay? And where does cellular respiration occur? Actually, it's going to be in your mitochondria of your plant, of your animal cells, which we'll be getting a little more specific. And lactic acid fermentation will occur in the cytoplasm of a plant or animal. So here are the reactants and products again. So this is what we're getting, what we're producing. So this one's lactic acid, this one's alcohol. This is what we're doing today. And how is it related to cellular respiration? Well, this is the opposite process, right? And this is, related to photosynthesis is the opposite process. And how is it related to cellular respiration? How is fermentation related? All living things use both processes to release energy. So the energy source for photosynthesis is what? What's the energy for photosynthesis? Sun. And what about for cellular respiration? What's the energy source? Yeah, food. That's what we're thinking about. And yes, you'd need oxygen too, but food. And then lastly, how about for fermentation? What it is? Again, it's food, particularly sugar molecules. So this last one here. So respiration, your raw materials are oxygen, sugar, and you're going to get water, carbon dioxide, and lastly, heat. Energy is going to be in the form of heat. This is why you are warm to the touch, because all of your cells are performing this chemical reaction quickly. So where in the cell does the first stage of respiration take place? Cytoplasm. You don't have to know all of these, but I'm going to show you for sure. So where in the cell does the second place? There's your mitochondria. So it starts in one, ends in the other. Again, this gets really picky. Fermentation differs from respiration because it doesn't require oxygen. Fermentation that occurs in yeast we're doing today is alcoholic fermentation. Which type occurs in people? Lactic acid in your muscles. And the process by which cells withdraw energy from glucose is called photosynthesis. No, it's not. It's called respiration. We don't do photosynthesis, right? We do withdraw energy from glucose, so we don't do photosynthesis. And then respiration is an energy releasing process that does not require oxygen. Is that true or false? False, right? That would be fermentation. Very good. Okay, so what I'm going to do is I'm going to go backwards to, I'm going to go back to the one video that I wanted to show you guys, and then I'll round up everybody else, and we're going to watch that other one together. before lab. So this is the one for you guys for right now. And this is actually a really, really nice representation of photosynthesis. I love it. It's very well done. So they've gone inside the plant cell here, right? Now we're inside. Those are, well, inside the plant leaf, and those are the cells, and we're about to go inside of a cell here. Oh, it's all graphics, but it's cool, right? So that's,
That must feel so special.
yep. So those green blobs are going to be the chloroplasts. So we're going to go into one of those. Isn't that cool though? And then those are the grana, right? The stacks. And then those thylakoids are those pancake things, right? And
The pancake.
yeah, that's where your actual reaction is taking place, your photosynthesis reaction. It's going to show you the actual molecules interacting right here. It's really cool. Somebody did a great job with this. Yeah, look at that. Mm-hmm. Isn't that neat? And where you see the flashing, that's electron transport. So that's little bits of energy popping here and there. That's pretty wild, isn't it? So I love that one. It's just good. All right, now we'll watch the last one together. And this one, Cole and James, y'all want to come? So this one's on cellular respiration, and it's good, very good. Amoeba sisters are great. You guys are going to have an entire lesson that's just amoeba sisters because a comparable lesson isn't in your book. Cole, you coming? He got his earbuds in. I want to strangle him sometime. Cole. Yo. Oh. All right, so this is cellular respiration. And then what we're going to be doing in lab today is fermentation. And I'm going to go get lab set up while this plays.
Are you a morning person? No. One of us is, and one of us is definitely yes. Mainly because when I wake up in the morning, it just takes a while for me to get my energy back. It takes a lot of time and coffee for that to happen for me. Cells really don't have that luxury. They are busy performing cell processes all the time. Active transport of many substances needed for their survival, for example. And the energy currency they need, specifically, is ATP. ATP stands for adenosine triphosphate. It's a type of nucleic acid, actually, and it is action-packed with 3 phosphates. We have a video all about ATP and how it works as an energy currency. So where am I going with this? Well, cells have to make this ATP. It doesn't really matter what kind of cell you are, prokaryote or eukaryote, you have to make ATP. The process for making that ATP can be different, however, depending on that type of cell. One way is called aerobic cellular respiration. Lots of organisms can do aerobic cellular respiration, but this video is specifically going to go into aerobic cellular respiration in eukaryotic cells. That means this video is talking about the processes in cells that have membrane-bound organelles, such as a nucleus and mitochondria. Eukaryotic cells include the cells you find in protists, fungi, animals, and plants. The mitochondria, which can be found in most eukaryotic cells, are going to be kind of a big deal in this aerobic cellular respiration because some of the process occurs in the mitochondria. So let's get started. Remember, the major goal for any organism performing this is to make ATP. Okay, here's the overall look at the equation for aerobic cellular respiration. Remember that reactants, inputs, are on the left side of the arrow, and products, outputs, are on the right side of the arrow. This equation, by the way, looks remarkably similar to photosynthesis. Look at how the reactants and products are on different sides. Well, that doesn't mean they're simply opposites. It does show that they have substances in common. In photosynthesis, organisms make glucose. Notice how glucose is a product. But in cellular respiration, organisms break the glucose down to make ATP. Fun fact, you know how when a bean seed first germinates in the ground, it isn't able to do photosynthesis yet? Yeah, the germinating bean is relying on glucose that it has stored, and it's doing cellular respiration to break it down to make ATP so it can grow. Of course, once it starts to mature and develop leaves, it can do photosynthesis. It will be able to do both photosynthesis and cellular respiration. Plants make glucose in photosynthesis, and they break it down in cellular respiration. They can do both. But if you aren't photosynthetic, such as a human or an amoeba, you have to find a food source to get your glucose. You need glucose to get this process started, and we're going to assume that we're starting with one glucose molecule. Step 1, glycolysis. This step takes place in the cytoplasm, and this step does not require oxygen. It's considered anaerobic. In glycolysis, glucose, the sugar from the equation, is converted into a more usable form called pyruvate. Glycolysis usually takes a little ATP itself to start up. The net yield from this step is approximately 2 pyruvate and 2 ATP molecules, and 2 NADH. What is NADH? NADH is a coenzyme, and it has the ability to transfer electrons, which will be very useful in making even more ATP later on. We'll get to that in a minute. Now, an intermediate step occurs. The 2 pyruvate are transported by active transport into the mitochondria, specifically the mitochondrial matrix. In the mitochondria, pyruvate is oxidized. The 2 pyruvate are converted to 2-acetyl-CoA, which will be used by the next step. Carbon dioxide is released and 2 NADH are produced. Step 2, the Krebs cycle, also called the citric acid cycle, still in the mitochondrial matrix. The citric acid cycle is considered an aerobic process. While the cycle doesn't directly consume oxygen, some of the events in the cycle need oxygen to continue. To learn more about this intricate cycle where the 2-acetyl-CoA will enter, check out the further reading suggestions in the video details. Carbon dioxide is released. We also produce 2 ATP, 6 NADH, and 2 FADH. FADH is also a coenzyme, like NADH, and it will also assist in transferring electrons to make even more ATP. Step 3, the electron transport chain in chemiosmosis. This is just a beautiful thing, really. In eukaryotic cells, this is still in the mitochondria, and to be more specific, it's involving the inner mitochondrial membrane. We do require oxygen for this aerobic step. This is a very complex process, and we are greatly simplifying it by saying that electrons are transferred from the NADH and FADH to protein complexes in electron carriers. The electrons are used to generate a proton gradient, as protons are pumped across to the intermembrane space. All these protons being pumped out into this intermembrane space generates an electrical and chemical gradient. The thing is, if you remember from our cell transport video, ions, like the H+, don't easily travel across membranes directly without something to travel through. The protons can travel through an amazing enzyme called ATP synthase. If I could be any enzyme, I'd be ATP synthase because it has the ability to make ATP by adding a phosphate to ADP. ADP is a precursor to ATP. ADP has 2 phosphates, but if it obtains a 3rd phosphate, it becomes ATP. So in chemiosmosis, the protons travel down their electrochemical gradient through a portion of the ATP synthase, powering it to make ATP, the ultimate goal, Oxygen is the final acceptor of the electrons. When oxygen combines with two hydrogens, you get H2O, water. If you remember from our equation, water is a listed product. Now, the electron transport chain in chemiosmosis step makes a lot more ATP compared to the other two previous steps. How much? So I've noticed in my years of teaching and the assortment of textbooks I've collected over the years, there are some different numbers in cellular respiration charts. In fact, you can even see them change a bit in different editions of the same book. And it's made me want to emphasize that it's important to not just memorize a number, because it really is more of a range. I want to focus less on the number of ATP made per glucose molecule because it depends on a lot of variables. One variable is the gradient we were talking about. How many protons were pumped across the mitochondrial membrane? You can see more variables discussed in the factual references. And those references have estimates ranging from 26 to 34 molecules of ATP per glucose molecule in the electron transport chain in chemiosmosis step alone. Then if you add the other two steps, Krebs, otherwise known as the citric acid cycle, and glycolysis, you could estimate a range of anywhere between 30 to 38 net ATP molecules total per molecule of glucose. Again, to emphasize a range. Now, this is just one way of creating ATP. But like we had said at the beginning, all cells have to make ATP, but the way they do it can differ. If there's no oxygen available, some cells have the ability to perform a process known as fermentation. It's not as efficient, but it can still make ATP when there isn't oxygen. We have a video about that. We can't emphasize enough how important the process of making ATP is for cells. For example, cyanide, which is found in some rat poisons, can block a step in the electron transport chain, which would block ATP production. A poison that prevents ATP from being produced can be deadly. With the important role that mitochondria have in ATP production, there is also a demand for increased research on mitochondrial diseases. We are confident that the understanding of how to treat these diseases will continue to improve as more people like you ask questions. Well, that's it for the Nuba sisters, and I remind you to stay curious.
Cool. All right, so what I'm going to do is I'm going to let the juniors that are online go, and I'm going to try and get a lot of video and pictures from today's lab, so I'll upload it. So you guys should be good. Now y'all can wait here a minute because we'll just go over some stuff before lab before we go in there. All right, so bye. See you later.
Bye.
Bye. Okay.